So first of all, thanks to the organizers for having me. It's really a pleasure to speak at this workshop and also to listen to all the other contributions. And I'm really looking forward to the coming 2 days as well. As was mentioned before, yes, I fit the session very well because I'm also using reconstitution methods and connecting a bit to the previous session, we are also looking at signaling processes with these reconstitution methods, specifically at how immune cells talk to each other. So this is what's depicted in the center. It is a model of clay made by one of my students. And the left is a T cell with like these long membrane protrusions interacting with an antigen presenting cell on the right. And as you can see in the background, we are using single molecule methods and also a reconstitution in membranes to study these signaling processes. So what's
I don't know whether it's just me, but I don't hear.
For some reason, I just muted myself. So a special part about immune cell signaling is that a lot happens in these cell-cell contacts. And on top of that, these contacts are also highly dynamic. And so what my lab is really interested in was going on in these interfaces at the molecular level. So how is the signal kind of transduced across the membrane and integrated in the receiving cell membrane? And yeah, as it was already mentioned, we use reconstitution, but I also want to motivate this a bit and put it in context. So of course, people have already used reconstitution methods for a very long time in the biophysics field, but also in biochemistry, where at least classically, people really study one molecule at a time. For example, looking at a reaction in a test tube, or also studying molecular interactions like with SPR, for example, on a chip to measure binding times and binding kinetics. But of course, this is really like a full reductionist view, which mostly looks at a component or maybe 2 or 3 at a time. And it also lacks the context in which these reactions normally occur. And on the other hand, of course, you can also study signaling in the context of a living cell. And this is also, of course, what we've seen in the previous talks, even in the context of tissues. And while this is extremely informative, of course, if you are interested in the molecular side of things, it also means that a lot of the processes get hidden by, because you can only ever look at maybe 1 molecule at a time or 2 or 3. And then all these other processes that still occur in the background can really influence your result, and you won't necessarily know what happens on the molecular scale. And so what we are trying to do is to move from the left to the right in a bottom-up biophysics approach. So still actually having individual components that are very well characterized, meaning purified proteins, model lipid membranes, but adding constraints and complexity by that I mean both controlling a bit more the spatial context in which the reactions happen and also the temporal context. And that is a bit like, I would say, a new direction there. So if we go back to this kind of picture of the 2 cells talking to each other, interacting with each other, ligand receptor interactions, then one model system that we've used a lot in the past, or I've used a lot in the past in my PhD and postdoc, is to look at cells interacting with model lipid bilayers that, for example, contain one of the ligands or other adhesion proteins, and then we can look at these cell model membrane contacts. So in this picture, we've essentially replaced one cell by a reconstituted system. And now what my lab is currently about to do is to essentially do this for both sides. So we have a fully reconstituted system, both for the receptor and the ligand part of the cell. So what can we learn by reconstituting kind of signaling processes in these really simple systems? You may think that you lose a lot, and that's of course true because we only ever focus at a few components at a time, but actually these systems can be already quite complex. So this is just a recent example from T-cell signaling where the Whale lab and Rosen lab reconstituted the first stages of receptor activation that happens on the inside of the cell membrane on motile-lipid bilayers. This was including 10 different proteins going all the way from the initial phosphorylation event to actin polymerization, and they could really find a lot about how these signaling processes organize in the membrane. It's definitely not random, and phase separation is a regulatory element to this. What you can also gain from reconstitution studies is kind of mechanistic detail about the order of events. For example, for that you don't necessarily even need single molecule resolution. So this is a really nice study on a completely different part of the immune system, innate immune system, where they essentially solved like a really long debate on how the different receptor parts have to come together to initiate signaling and what is the order of events. And finally, going a bit more towards single molecule methods, we can really zoom in on the reaction kinetics and the mechanism. And for example, this study is a very nice example of that, where they showed that a very important molecule in T cell signaling, which also has a very high relevance to cancer processes actually, is actually governed by kinetic proofreading mechanism. So this ensures that the molecule only activates when it should, because this process needs to be so tightly regulated, and all these details would be impossible to obtain from measurements in live cells just for the technical difficulties. So the system that my lab is currently studying and interested in is interleukin signaling, more specifically interleukin 2 and 15 signaling. And just to give you some idea of what the signaling pathway is about, they are key regulators of the immune system. And the two interleukins that we want to study play, for example, a role in the recognition and defense against degenerated body cells. For example, when a cell has some mutations and is on its way to become a cancer cell. So two things can happen in that case. All body cells normally have a kind of passport molecule that signals to the outside that they are normal, which is the MHC complex 1, and it can present peptides derived from the cell. And in a normal case, the cell would be ignored. But if the cell presents abnormal peptides from a mutated protein, for example, then so-called killer T cells or CD8 T cells can come recognize this and destroy the cell. But alternatively, a mutation might mean that this passport molecule is downregulated, and that is called a missing self event. So this cell now doesn't show this molecule on the cell surface. And there's another group of immune cells called natural killer cells, which can recognize this situation and then also lead to killing of these cells. Now this, in principle, the slide could maybe stop here, but in reality, the situation is more complicated because these initial events would normally not be enough to really have a sustained response against these cells, and most likely there's not more than a single cell. And so what happens is that as these cells get killed, so-called dendritic cells take up fragments from the killed cells and present it on their surface. And as they do so, they enter an activated state, meaning they present also interleukin 15 on their cell surface. And now in a kind of second round of immune cell recognition, we get helper T cells that recognize these kind of abnormal fragments, and NK cells that get activated by interleukin 15 in this case. So this very molecule that we are very interested in. And then that leads to expansion of both NK cells and T cells. And because of that, and because this is clonally selective, we really get a very specifically tailored response against this particular cancer cell, or it could also be a cell infected by a virus. And now, interestingly, for this proliferation of the T cells, we need interleukin 2 signaling. So this is a signaling molecule released by T cells to stimulate their own growth. And what's special about this interleukin 2 and interleukin 15, you can see that they have very different roles in this kind of process, although they are both really important to, as I said, get this sustained immune response. But molecularly, they are actually extremely similar. So if we zoom in on what the receptor looks like, they actually share the two signaling subunits. So both the receptor for interleukin-2 and for interleukin-15 has a gamma and a delta, like a beta and a gamma subunit that are identical. And then they have a specific subunit, alpha subunit, but this one is actually not involved in the signaling process at all. So as the ligands bind, this is a classic example of where essentially signaling is induced by introducing proximity between bound kinases, the JAK kinases shown here. So the receptor gets phosphorylated, downstream signaling molecules like STAT5 can bind, and this leads to overall downstream signaling. Now, biophysically, of course, the question is, if these are really identical, how can the cell actually know whether there's interleukin-2 or interleukin-15 bound? Since essentially the intracellular processes are, of course, to them, what happens on the external side is invisible. And even though these molecules are so similar and they sort of are present in similar types of immune responses, they actually introduce different features in the cells. So it is really not that one can be completely exchanged for the other. So leading to the question, how do these two molecules produce distinct signals using the same pair of signaling subunits? Now, of course, the very classic picture you may think of immediately is conformational change. So maybe these ligands just introduce a different conformational change leading to a bias of the phosphorylation pattern. But actually, they have a quasi-identical atomic structure in the ligand-bound state. So this is an overlay of the two crystal structures of the receptor, one time bound with interleukin-2 and one with interleukin-15. These are the green and the purple molecules in the center. And you can really see that the two receptor subunits are positioned kind of identically. So this seems really unlikely. Now, of course, like if you look at what has to happen in terms of the signaling process at the cell membrane, we can see that all these molecules have to come together. So we have a 2D component of the receptors moving in the membrane and then the ligand binding in 3D. And so this led us to wonder whether the assembly kinetics could simply be different for the different ligands. And then, of course, mechanisms like kinetic proofreading can really lead to different phosphorylation patterns depending on different off rates, for example, of the foam complexes. Another interesting thing is that space could also contribute to discrimination in this case, because the reaction geometry is actually different for interleukin-2 and interleukin-15. So in one case, the ligand is presented by another cell in a cell-cell contact. That's the activation of these NK cells by dendritic cells by interleukin-15. And in the other case, interleukin-2 is more soluble signaling molecule secreted from cells to activate other cells. So again, this kind of different situation could lead to, in the end, differences in the kinetics of the assembly of the receptor and by that signaling processes. So in order to test this hypothesis, we are working on a reconstituted system of these interleukin receptors in model membranes, both the case where we have a soluble ligand and also like a ligand presented by another cell, so in a bilayer interface. And this is a project led by my PhD student, Christian. And for that, of course, we first have to make these proteins and also the membranes. We had to build a microscope for the single molecule measurements, and then hopefully we can answer these questions that I just outlaid before. So on the first step, because these proteins are actually quite complicated to make, we have to make them in insect cells, and also labeling them, we had to devise a method that really ensures that we have one-to-one stoichiometry and a very high degree of labeling. And for that, we resorted to an enzymatic reaction based on the enzyme sortase, which can essentially attach a peptide to the N-terminus of a protein very specifically. And with that, we get a really high amount of labeled. We can do a kind of two-step purification where once we purify from one end and one from the other end, so we really only end up with labeled protein. And then we can reconstitute these proteins in the membranes. And currently we do this simply by his-tag binding to nickelated lipids. So we actually only use the ectodomain of the receptor, so the part of the receptor that's extracellular. And as for the microscope, I just have one slide on this because we really spent quite a lot of time trying to build the optimal setup with the like kind of state-of-the-art components, but also for the cheapest money possible. And really trying to make a lot of things in-house. For example, we don't use a camera body anymore because this is quite expensive and not very flexible in terms of adding, for example, different dichroic mirrors. So we based this on a design called MyCube from the Holbein group in Wageningen. And if anyone is interested in this in more detail or maybe needs to have a new microscope for their own lab, we actually have now an own website for this microscope where we very openly share all the components and instructions on how to build a microscope like this. And I should say that we really have simultaneous 3-color detection. And with the new CMOS cameras, actually also over quite a large field of view. And we use a beam shaper to get rid of a Gaussian illumination profile. So we have a flat top illumination. And this is just a video showing the very first data we got with these, combining this protein labeling approach with our new microscope setup. So this is just a model experiment where we essentially follow the association of an antibody to a ligand-bound receptor in the membrane. And by following their diffusion over time in 2 colors, we can follow also their association. And of course, in the end, we will extract kinetics binding times from this data. So that's future work that still remains to be done. But as often, from a theoretical point of view, if you do theory, you can sometimes get results a lot faster. So I also want to present a few results that we obtained with Peter Rein, in collaboration with Peter Rein, Ten Wolde's group, who is also actually in this meeting. And so they have developed this really nice approach to simulate reaction kinetics into the based on a Green function reaction dynamics. And so we can really simulate like essentially the behavior of this receptor system completely in silico, and really follow which species essentially are present depending on the ratio of input. And thankfully there's also already a lot of kinetics measurements there that are all based on 3D kinetics, but they give us a first starting point. And so from these previous 3D kinetic measurements, so by that I mean really measurements in solution, so like the molecules are not bound to a membrane, so that's actually quite a difference to our system, and that's also partially why we want to do the measurements that we do. People have already measured, of course, the affinity of the ligand, for example, to the alpha chain, or to a beta and gamma chain, or the whole complex together. And from that, they got some estimate for the prevalence of these complexes. And so what the field has assumed up to now is that the alpha chain, essentially, which is not involved in the signaling, acts as a sort of scavenging receptor that captures IL-2 from solution and then brings it to the beta and gamma chains in the membrane. So because this affinity is actually in the solution measurements pretty high. But when we did, or when I should rather say, when Peter Rein's student did the simulations, we found that actually this particular fraction is non-existent because of the fact that actually all these molecules are membrane bound. There, of course, the interactions in the membrane kind of dominate over the interaction from solution. So it essentially what happens is that this kind of complex is much more prevalent than it was originally predicted, simply because the interaction between beta and gamma is stronger since they're both membrane bound than you would estimate just from their solution kinetics. And everything that binds to alpha is essentially immediately transferred to the full receptor rather than just alpha interleukin 2 being a species on their own. And this is really interesting because it could potentially explain why blocking the interleukin 2 alpha complex has had limited success in the clinic. So this has been a major drug target. But if essentially most of the time interleukin 2 can bind to beta and gamma also in the absence of alpha, then this kind of can explain very nicely why blocking this interaction is maybe not so effective at disturbing interleukin 2 signaling. So jumping a bit from the, like one reason that these experiments are actually tricky to do or one reason that extracting kinetics from single molecule experiments is actually rather tricky is that, of course, if we use single fluorophores as labels, we have a limited observation time window as these fluorophores bleach. And this also typically limits our time scales to like several seconds or like tens of seconds at the very best. Now, we have also, in a collaboration with the Schiller Lab at the Max Planck Institute for Biochemistry in Martin Street, looked at ways to overcome this issue by actually getting inspired by a new super resolution imaging method that's called DNA Paint. And we repurpose it for single particle tracking. And the idea is that instead of labeling our target molecule with a single fluorophore, we actually label it with a single-stranded DNA molecule. And we have so-called imager DNAs in solution, which are short DNA molecules that are complementary to the long DNA strand that we attach to the target molecule, and they all carry a fluorophore. And now if we essentially tune the kinetics so that the continuous turnover happens on a time scale that is similar to the bleaching rate of the fluorophore, we in principle should be able to image our molecule for an infinite amount of time as bleached labels get continuously exchanged with solution. And this really works very nicely. So this is just some data where we use the same dye. It is actually in this image still a DNA origami statically attached to a cover slip. And in one case, we label with a single dye, and we get typical observation times of 20 to 30 seconds before the dye molecule bleached. And you can see after 2 minutes, there's hardly anything left. Whereas if we label with this new DNA paint approach, we can actually keep observing molecules for half an hour, or even in extreme cases for more than an hour, until we lose them. And we can also make this a bit more quantitative by looking at the particle, the different trajectories we get per particle. And so we cannot simply just compare times, because actually it's not just the times that change, the observation times for an individual molecule, but also the number of times we observe a single molecule can change. And that is because we can have stretches where we don't see a molecule because all the DNAs have detached or bleached, but then they can reattach. So we can see the same molecule over like a given window of time. We can see the same molecule multiple times for multiple lengths of times as well. So in order to have the fairest comparison, we came up with a measure of trajectories per particle that we can see. And if you make a distribution of the number of trajectories, these trajectories that are longer than a certain time t, then you get a certain distribution for the single dye. In this case, as I said, the typical half-life of these trajectories on the order of a second, so that fits very well with like what we typically get from single molecule measurements. And of course, it starts at 1, because if the single dye is bleached, then we can't see the molecule anymore. And for the tracking handle, we can see that on average we get about 20 to 30 trajectories per particle, And the average length of time that we can observe them for is well over 1,000 seconds. So quite considerable improvement over the single dye case. In fact, depending on the power we use to image, and that affects, of course, the bleaching rate, we get a 20 to 30-fold improvement of observation times for this new method. Last, it's also a bit in the sticking to the theme of methods to really explore these systems further in the future. Of course, these signaling processes don't happen in an open system normally. So at the moment, we study all these processes on supported lipid bilayers. So we have an open reservoir system. But of course, in the end, what we would be really interested in doing is studying them in a closed system, like a vesicle, for example, where we also have like realistic constraints also in terms of concentrations and molecular numbers that are more similar to a live cell. And for that, we are also really working actively on methods to make lipid vesicles. And it's also in collaboration with people from the BASIC project in the Netherlands, for building a synthetic cell initiative, where the idea is to really reconstitute like a dividing cell from the bottom up. And the particular type of method that we have really spent a lot of time working with over the last years and essentially improving is called continuous droplet interface crossing encapsulation. And the name kind of already describes what happens or how this method works. So we make liposomes by essentially spinning like a disc of a macroscopic size of several centimeters, and we first add an aqueous phase, which is going to be the phase outside the vesicle in the end, and then we add a layer of oil that contains also lipids, and in the end we insert a capillary into this spinning rotating chamber, with which we can inject the solution that we would like inside our vesicles into this oil phase. And so what happens is that we first get a formation of micelles in the oil, and as they pass the second interface, we get a formation of a bilayer and a formation of vesicles. And of course, the centrifugational force essentially ensures that these particles or the vesicles travel from the inner side of the disc to the outer side. And this is what the setup looks like in real life. And we also have the plans on our GitHub website if anyone is interested. So what we tested was whether the diameter of this capillary that we use will make a difference on the size of the vesicles. And the reason we use so large capillaries, so they are much larger than the vesicle size that we expect. So we get vesicles, as you can see, like of around 10 micrometer diameter. But if you want to encapsulate viscous protein solutions using capillaries that are that small and that then they really get clogged all the time. So this was a major bottleneck actually. So but if you go to larger capillary sizes, what you lose is a bit of the control of the size. So now our vesicles distributions look really almost identical no matter which size we use, but we get very high numbers of vesicles in the thousands and can really do nice statistics on these vesicles. And so essentially there's no particular benefit anymore to go to particularly small sizes, so we can work with 100 micrometer, which essentially is really, really convenient for the experiments. What was a problem or has been a problem and why the field has not really adopted this method so much in the past, I think at least, is that the reliability of the method has been pretty bad. So yeah, people would get good results one day and then the next day maybe you wouldn't get any results, and that is something that really also upset me in my postdoc time actually, that people had such little understanding of why this method was so unreliable, or similar methods as well. So we spent a little bit of time looking in environmental factors. And one thing that people speculated a lot about before, but nobody had really shown, is that humidity actually makes a big difference, because we have this kind of two-phase system. So, and the oils and lipids are a bit hygroscopic. So if you have a high humidity, then essentially you change this, you change the behavior of your two-phase system quite drastically. So what we could find is that actually if we have a humidity of already 60% or so, then we get considerably smaller vesicles and also sometimes no vesicles at all. And this gets even worse for really high relative humidities. And this is actually, in the Netherlands at least, could be quite realistic for the humidity in your lab. So, so we actually now use a dehumidifier, and we also prepare our oil mixtures in a glove box to control this parameter better. And I've really seen this make a big difference for the reproducibility, also across 3 different labs in the Netherlands at this point. And another factor is that this lipid and oil mixture, which also contains actually chloroform, is pretty unstable. So it spontaneously demixes, and the lipids also can crash out of solution again, particularly if you leave it open, and some of the solvents can evaporate. So this also, and this is a time scale that you may not actually consider. So, I mean, it's quite short. So people, if you don't really know, then you may well not think that you have to actually work that fast in order to assure that that you actually use the same solution at all times. So it's also kind of a time critical experiment. So with that, actually we could use this method for a variety of applications, and it works really very well. We can show that the membranes are unilamellar and that they are oil-free, which is something that's very important if you want to later insert proteins into them. And we can reconstitute an actin cortex inside these vesicles. That was done in collaboration with the group of Gijsje Koenderink in Delft. We can also reconstitute a synthetic cortex based on DNA origami structures inside these vesicles. And we can also encapsulate vesicles and vesicles as a first step towards creating a system that shows some multi-compartmentalization. So far, so really like anything we try to put inside these vesicles has also ended up inside the vesicles, which is sounds trivial, but is pretty exciting for people who've been working with this for some time. So with that, I really want to thank my group for like the last 2 years. It's been really a fantastic journey. And of course, similarly, all our collaborators, Peter Rhein, but also Gijsje Koenderink, Kees Dekker, Christophe Danelon, who really helped with getting these projects off the ground. And you for your attention. Thank you.
Thank you, Christina. That was wonderful. Questions from the audience? I'm sure there are some questions from people that are much smarter than me.
Oops.
Personally, I'm not an expert in the immune system and this type of work, but so you've sort of shown a lot of technology in that you're developing. Sort of, could you maybe give a bit of some ideas of what you would like to see or what you expect to see if once you actually measure sort of this ligand receptor dynamics in time and space? What are the type of things that you hope to find or you expect to find?
Yes, yeah. So
it's maybe a broad question, but you can sort of then pick a bit what you prefer to talk about.
Yes, sure. Yeah, so basically, yes, as you say, I mean, we've been really busy just setting things up for the last 2 years. I think that that with this methods in place, we can really start looking at how information is integrated by receptors. So this was just a really simple example to start with the interleukin signaling, where it's essentially just about kinetics and whether they are different between the different scenarios and how this links to their biological function and ligand discrimination. But for example, one other receptor that I've worked on a lot before and that I would like to continue working on in the future is the T-cell receptor, which is a much more complicated system in the sense that it's not just simply a yes or no decision, and discrimination becomes really like a much more interesting question, because there's so many different peptides, for example, that have been discriminated have to be discriminated and very reliably and on very fast timescales. So using similar approach, we could hopefully also look a bit more into the different mathematical models that have been proposed for this, and really try to experimentally test them in a system where we can really measure all the parameters and also know all the components. Mm-hmm. And obviously also like going towards downstream signaling, I mean, turning this around, not just looking at what happens on the extracellular side, but also what happens on the intracellular side. Mm-hmm. Is also will also be an interesting direction.
Got a question from Jordi? Go ahead.
Yes, thank you so much, Christina. I have a question. You mentioned that there is a difference between IL-2 and IL-15 in terms of one is diffusible and the other one isn't. And I was wondering what type of, what differences would you ascribe to this different behavior given that the cells are moving around? So I'm curious to know, is there anything that you can assign to this different behavior of the two cytokines regarding the behavior of the system?
I'm not quite sure I understand your question perfectly, but...
Well, what I mean is that since the cells are moving around, maybe I would mm-hmm. expect not too many differences from the fact that one of the cytokines is diffusing and the other one is bound to the membrane.
I see, yes. Yeah, so basically I think that is of course true that like if you just think of the cells moving around in the lymph nodes or also in the body, that there could be situations where just one is not present anyway and only the other one is present. But actually, in fact, if you have a full-on immune response, then you will have at a very similar site of infection or in the lymph node, which is quite a crowded environment, you will have secretion of this interleukin-2 molecule in parallel to this the need of having this specific recognition event in a cell-cell context for interleukin-15. And so there definitely will be the situation where both will be present, and it will be important for the cell to discriminate between the two events. So for the NK cell in particular, of course. There's also a more limited case to make for the fact that interleukin-15 is shed off the cells as well. And there's also these micro vesicles released by immune cells, which are now actually looked at a lot also in the context of immunotherapies. And so there could also be, the reverse could also be happening, that you have interleukin-15 being presented to T cells that should in principle respond to interleukin-2.
Okay, thank you. Thank you so much.
Next, we had a question from Henny Valkenier.
Thank you very much for this talk. And I was very interested in seeing indeed the different methods that you develop, and in particular the method to make giant unilamellar vesicles. Mm-hmm. For that, I have two questions. First, are there still traces of oil left in the membrane when you make these GUVs by this C-DICE method? And secondly, what lipids can be used? Do certain lipids work better than others, or is it quite universal?
Yeah, so that's a good, both are good questions. So first question first. So regarding the residual amount of oil. So the two things that we've done in our lab directly is this reconstitution of hemolysin, and it is known to be quite sensitive to the dimension of the bilayer because it has a very limited tolerance for how far the lipids can be spaced apart. So if this works this works really very well, and is kind of gives us an indirect readout of there cannot be a large layer of oil between the two leaflets. Of course, this doesn't rule out that there could be like traces left of individual oil molecules. And then we've also done essentially like fluorescence measurements showing that that we don't actually have more intensity than we expect for the membrane, which is also like a good control to have. But there are also other groups who have looked at the mechanical properties of these vesicles, and by fluctuation microscopy methods, and also by pulling methods. And in both cases, they have actually found them to be not significantly different from electroformed GUVs, which is also very confidence inducing, because if you have a large oil reservoir, then the mechanical response will be very different. And as for the second part with the different lipids, for sure there will be a difference between different lipids. We see that. But we can, for example, even get quite high amounts of pegylated lipids in our membranes, even though like you would maybe first expect that that is a bit of an issue with the oil phase. So it turns out that solubilizing the lipids in this oil, like getting that step right, is really crucial. And now we think we have a very good handle on this. It's not, I would never say it's completely universal, but it's quite universal in our hands. So we tried already like very many lipids, even charged lipids. And so far anyway, we haven't run into big difficulties. Maybe I mean, cholesterol we haven't tried yet. So this is still a question mark, and maybe the biggest one for people working with membranes. So that remains to be seen how well that works. But I have some hope.
Thank you very much for these informations and for this nice talk. Thank you.
I think we're exactly times right. So thanks again, Christina, for a wonderful talk.